Okay, hello everybody. In this video lecture, we will cover neuron physiology. So let's go ahead and begin. And when I'm done, I'll see if you guys have any questions. I will be happy to answer them. So before talking about neuron physiology, this is just a very basic, very quick review of electricity, actually principles of electricity. And what is electrical potential? Electrical potential that can be measured in voltage is actually separation of opposite electrical charges. If opposite charges are separated, the system has potential energy. So example would be just a regular battery that you use every day, right? If you look at a battery, you see they have a positive end and negative end. That means that we're separating those charges. And because those charges are separated, this battery has voltage. It means it has some potential energy, right? So that's our electrical potential or voltage. What is electrical current? Electrical current is a movement of electrical charges, right? So also, if we take example a battery, right? So we can see when electrons are moving, right, through the wire from negative to positive end of a battery, we can convert this potential energy into another form of energy, right? So we can your flashlight can work, your remote control can work, your computer works, right? So when charges are separated, that create potential energy. And when those charges start moving, we create electrical current, all right? So now how it is relative to physiology of neuron? Because actually not just neuron, but all cells of your body establish this electrical potential. That means all cells of your body separate positive and negative charges. And this electrical potential called resting cell membrane potential, and it can be measured, and it's measured between 70 millivolts to 90 millivolts, right? So every single cell of your body is a little battery because your cells separate positive and negative charges. And because of this separation, this potential energy is stored, and this potential energy can do some work within your cells. Now, what is the function of neurons? So the function of neurons is to conduct an impulse. And it can be excitatory impulse, it can be inhibitory impulse, but that impulse is a movement of electrical charges. We have two types of conduction, normal conduction and saltatory conduction or propagation, and we will talk about it a little bit later in this chapter. But if you look at this diagram of a neuron, so I'm just reminding you that neuron has a neural body, right, and dendrites, and then human neurons, they have one axon. So axon carries information away from the cell body. And this information is in a form of electrical impulse. So if you look at neuron, right, so here's this little tube represent neuron. Actually, all our cells and soma and dendrites and axons, they have negative charge inside. So they have negative charges inside and positive outside. But now, when these charges start moving, right, you can see how positive charges moving in, and axon became positive inside. This is movement of electrical charges. This is electricity, and this is electrical impulse, and that's a function of neuron to conduct this electrical impulse. And your nervous system understand those impulses as some message, right? So this is how your body communicates. Different parts of your body communicates, and your body communicates with external environment, right, through this electrical and some chemical impulses, chemical messengers and electrical impulses. Now, what are excitable cells? Excitable cells are cells that are able to produce and respond to electrical signals. And those excitable cells would be neurons, muscles, including all types of muscles, skeletal muscles, cardiac muscles, and smooth muscles, and secretory cells, such as your endocrine gland, for example. So what do we need to do for cell to respond to these electrical signals? Or how your neurons actually able to create this electrical impulse, right? I just told you that function of your neuron is to conduct this impulse, but how this impulse is generated, right? So first, your cells need to separate charges. And this is done, the separation of charges is called electrochemical gradient. And when your cells separate charges inside and outside a cell, creating this electrochemical gradient, this creates potential energy. Now, to separate charges, your cells require several membrane proteins, right, to create this charge separation. So let's look what proteins are involved, and those are membrane proteins. So here's the membrane proteins that are involved in separation of charges. So just look at this like that. You are trying to create, let me see. So you're trying to separate charges inside and outside of a cell to create potential energy, to make your cells a little batteries, right? To make your cells little batteries. So here's the membrane proteins that are involved. Sodium potassium ATPase, another name is sodium potassium pump. Sodium potassium leakage channels, sodium calcium potassium voltage gated channels, mechanically gated channels, and ligand-gated channels. So all these channels are involved in separation of charges. Now this is sodium potassium pump. All pumps, they require ATP energy to do their work. And so you can see on this diagram, we have cell membrane, phospholipid bilayer, and here are our transmembrane proteins, right? This specific protein is sodium potassium pump. So what it has binding site for sodium, and it has binding site for potassium, and also another binding site for ATP, right? So for this phosphate group, right? So ATP donate its phosphate group to this channel, right? It became phosphorylated, and then, you know, when this happened, this pump pushes sodium out and potassium in the cell, right? So sodium binds, right? 3 sodium binds from cytosol, so from inside the cell. Then we have phosphorylation. So ATP is used. So here we have phosphorylation of this channel. Now sodium is released, and then potassium binds, right? We have dephosphorylation, this phosphate group removed, and then potassium is released into cytosol. So sodium potassium pump using ATP energy to pump sodium out, potassium in, sodium out, potassium in, out of a cell, inside a cell, right? Sodium always out, potassium in. This is the job of the sodium potassium pump. Every single cell in your body has it. Another type of channels that are needed, sodium potassium leakage channels. So leakage channels, you know, it's easy really. Those are channels that always open, and those ions just go down their concentration gradient, right? So they are leaking out of these channels. Because phospholipid bilayer over here, right? It doesn't allow charged particle to move through. So they need, you know, channels to do it. So here you can see ions from high concentrations, they move down their concentration, right? Like pretty much through diffusion. This is called facilitated diffusion because they do move down the concentration gradient without ATP, but they use channel to facilitate this process. So that's facilitated diffusion through leakage channels. Now those channels are specific. If it's sodium leakage channel, then only sodium can move through. If it's potassium leakage channel, only potassium will move through. Another channels that are needed are sodium, potassium, and calcium voltage-gated channels. Now, voltage-gated channels means that they are closed, right, when cell is in a resting stage. So when cell is resting, actually cell inside is negative, outside is positive, and this voltage about minus 70 millivolt. So this is resting cell, and channel is closed. Right now, when voltage is changing, so over here you can see that voltage now minus 50 millivolts. Now this channel opens and it's allow those ions to move down their concentration gradient. So they again moving down their concentration gradient, but only when we have change in voltage, right? And they also specific. Sodium moves through sodium voltage-gated channel, potassium through potassium, calcium through potassium VGC, right? Mechanically gated channels. Mechanically gated channels when we have a protein channel and it's also closed, right? But then we need some physical, you know, kind of like disturbance of this channel. And then this gate opens. So some mechanical force need to be applied to open this gate. And again, sodium, potassium ions will move down their concentration gradient. For example, would be, let's say if somebody touches you. If somebody touches your hand, touches your skin, right? This is physical, you know, process, right? So this some kind of mechanical, you know, physical energy, right? And that when somebody touches your skin, it's disturbed these mechanically gated channels. And those channels open and we start seeing movement of ions, right? And ions, again, they move down their concentration gradient. All we need to do here to open those gates. And ligand-gated channels, you can already guess, should be some chemical. Any chemical that binds to a channel is called ligand. It can be neurotransmitter, it can be hormone, but when this example is neurotransmitter, acetylcholine, when it binds to channel open, allow again sodium, potassium, calcium, right, depends what kind of channel is that, move down its concentration gradient, right? So, so why are we talking about it? Because we said over here that all cells, and specifically neurons, because we will talk about neurons in this chapter, they separate charges. Negatively charged ions and molecules are inside, positively charged are outside, right? So we can measure this voltage, and this will be about minus 70 millivolt, and this is called resting membrane potential, cell membrane potential. And now, well, but how do they separate those charges? So for physiology, it's not enough to know that charges are separated. We need to know how are these charges separated. And that we said, okay, well, membrane proteins are involved that we call channels. What specific channels are involved in separation of charges? Sodium, potassium pump, sodium, potassium leakage channels, sodium, potassium, calcium, voltage-gated channels, mechanically gated channels, ligand-gated channels, right? So those are all different channels that involve in separation of charges. Okay, so now your cells, including your muscle and neurons, they establish resting membrane potential Now, resting membrane potential is a graded potential. We already talked about graded potential in the previous chapters. And graded potential has many names, like receptor potential, generator potential, postsynaptic potential, right? So that's a graded potential. In some, like axons and your neurons, they can also establish action potential, right? So all cells establish resting membrane potential, right? Many cells establish graded potential, and some cells establish what we call action potential. So this is 3 different potentials, but all of them involve separation of charges. So resting potential for all cells. Now, so here you can see, well, let's say this is a dendrite, right? Or it can be axon. It doesn't really matter. But you can see how inside the cell we have negative charges and outside we have positive charges. And when we measure it, it will be minus 70 millivolts across the membrane, right? But how, so this is resting potential, right? So that's your battery, right? So this is battery that's ready to work, but it's, you know, right now it's resting. Now this is established by sodium-potassium pump. So this pump constantly, remember what it does? It's pumping sodium out, potassium in, sodium out, potassium in. Now look, if you're constantly pumping sodium out, right? Wouldn't you expect to have high concentration of sodium outside, right? So here's the concentration of sodium outside, 150 millimoles. And what is concentration of sodium inside? 15 millivolts, right? So this is concentration gradient for sodium, right? So this sodium-potassium pump constantly pumping sodium out, potassium in. That's why we have lots of sodium out and we actually have lots of sodium or potassium in, right? So sodium, potassium. So your cells are salty bananas, right? Salty sodium, potassium. So that's a chemical gradient. But look, sodium, potassium, they both are positively charged. So if we pumping some positive particles out and some positive particles in, we don't really separate charges, right? Now, what's gonna happen? How are we gonna separate charges? That's why we need these leakage channels. We have both leakage channels for sodium and for potassium, but for potassium, we have, well, the cell membrane is 75 times more permeable to potassium than it's permeable to sodium. So we can, for, you know, for a purpose to understanding, we can ignore that sodium leakage channels. Let's just say that we have potassium leakage channels. Now, when we pump lots of potassium in, right? We have high concentration of potassium inside and we have leaky channels. So obviously potassium start leaking out down its concentration gradient. Now look, potassium leaking out, we're losing positive charges. And when we're losing positive charges, right, cell became more and more negative inside, right? So this is all chemical gradient, right? Sodium-potassium pump separates sodium and potassium, creating chemical gradient for sodium and for potassium. Because we have potassium leakage channels, potassium, that we have the whole bunch of potassium inside, start leaking out, right? And this start creating electrical gradient because we're losing positive charges from inside the cell. Right, so when potassium positive charges leaving, we became negative and negative inside. At some point of time, chemical gradient and electrical gradient became equal, and then cell established this resting membrane potential. So potassium leaking out, but potassium can also come back through the same channels. Why would it come back? Because negative and positive charges attract. So potassium moving out, some potassium moving in. When this movement became equals, that's the resting potential is done, right? Minus 70 millivolts. So now your cell became a battery. Okay, now this is resting potential for all cells. Now graded potential. Graded potential for muscle cells and for neuron soma and dendrites as well. Now graded potential occur when a stimulus causes gated ion channels to open. Magnitude varies directly, it's graded with stimulus strength, and it decreases in magnitude with distance, so it's short distance signals. So if you look at this picture down below, now what do we have? We have this axon, right? This is axon terminal, and this is another neuron, so that's a soma of another neuron. Now, we have electrical impulse going down this axon. When that happens, neurotransmitter is released, right? And neurotransmitter opens sodium-gated ion channels, right? This is gated ion channels. So those are ligand-gated ion channels. And when those channels are open, sodium start moving in. So, right, the positive charges start moving in. That's already what? Electrical impulse. Because remember, what is electricity? It's a movement of electrically charged particles. Sodium is ion, so sodium start moving in. So that's some type of electrical impulse, and it's graded potential because it's decrease in magnitude and it's just really short distance signal. And the last potential that cell establish, and those are muscle cells and neuron axons, is action potential. Now, action potential happens. So let's look over here on. Okay, let's see. So, you know, your cells are negative inside, right, and positive outside. So that would be a normal situation. So in some cells, in muscle cells, and in axons, sometimes we have reverse of charges. We have positive inside and negative outside, and this is action potential, right? So action potential involved voltage-gated channels, sodium, potassium, calcium. Now, when voltage changes, right, then sodium moves in, right, and cell became positive inside, and then potassium moving out, again, when voltage changes, so cell return back to normal stage, right? So it's a reverse of resting potential. It's called action potential. So yeah, it says over here, so let's read it. So action potential is a brief reversal of membrane potential with a total amplitude of about 100 millivolts. Occurs in muscle cells and axons of neurons, does not decrease in magnitude over distance, and it's a principal means of long distance neural communication. So let's go back and review what we are talking about over here. So we said that your cells can establish potential. What is potential again? Well, potential is separation of opposite electrical charges. So your cells can establish potential. What kind of potential? Well, we have three different types of potential. We have resting membrane potential, we have graded potential, and action potential. Now, resting membrane potential in all cells. And this is done because of electrochemical gradient, because sodium and potassium separated first, then potassium start moving out of a cell, making cell negative inside and positive outside. So that's resting membrane potential. And it's measured from -70 to -100. So it can be -70, -80, -90, -100 millivolts. That's a resting potential. What is graded potential? Well, graded potential, first, it happened not in every cell, but only in muscle cells and neurons, soma, and dendrites, right? Not in axons. And this has happened when we have ligand-gated ion channel open or mechanically gated ion channels open. But those are gated channels that need to be open. When gated channels open, we have movement of ions inside a cell, like here example, sodium moves in, and this is graded potential. And it decreases in magnitude, and it's a short distance signal. And the last potential that cells can establish, muscle cells and axon, is action potential. And for action potential, we need voltage-gated channels, right? When voltage is changing, right, and ions start moving in and out of a cell, right, that's electrical impulse that moves along axons, right? That's an action potential. And action potential is a reversal, complete reversal of potential from minus inside to plus inside a cell. It only occurs in muscle cells and axons of neurons. It does not decrease in magnitude. And this is how your neurons communicate. It's a long distance neural communication through this action potential. Right, so here let's look at the how that would look on a graph. Now that's a, very good pictures over here. So this is a membrane, cell membrane. Inside is cytoplasm and outside is extracellular fluid, right? So we have intracellular fluid, extracellular fluid, and we have cell membrane that separates, right, cell from its external environment. So phospholipid bilayer here. Now look, it is the local. So it does not have the beginning over here -70, but if you move a little bit before this point, right? Before this point, we have -70 inside, right? So here's -70 inside. This is called resting membrane potential, right? Now, and where we at? We are right over here at the beginning of axon. Now, when we have a local potential that comes from the soma, a local potential, what it does, it actually allow sodium in. So sodium start. So let's say this axon is excited, right? So let's say, let's imagine that you see those dendrites over here, right? Let's imagine that this axon is in your skin, in your, I don't know, elbow, right? So that's, you have your skin that cover your elbow, and in that skin you have this, well, it doesn't look like that, right? Because it needs to be bipolar. But just for purpose of understanding, right? Let's say that somebody touching you over here, and when somebody touches you, mechanically gated channels open and sodium start moving inside this neuron, right? So sodium moving in, sodium moving in, sodium moving in. So before, this neuron was resting, so it was -70. Now, if we have some either, chemical attaches or maybe some mechanical, some mechanical force applied, but something that open channels, ligand-gated channels, and sodium start moving in. When sodium start moving in, sodium is positively charged, right? So inside a cell became less negative. So we're moving towards 0, right? So we're moving from -70 to -60 to -55, right? So, and so you see how we're moving more and more positive inside because of local potential. Now, when it reaches -55, the voltage reaches -55, that is called threshold, voltage-gated sodium channel open, right? So voltage-gated sodium channel open over here, and sodium start moving in. So actually -70 is right here. You see where we have minus here? and plus over here, right? So that's all minus 70. So that's a resting membrane. Minus, plus, minus 70. So this is resting membrane. Now when local potential moving towards this channel, you see how it became positive over here? So we're moving toward 0, and when voltage reaches minus 55, these voltage gated sodium channels are activated and sodium enters axon. Sodium start moving in. Positive charges moving in. What happened inside the cell? It became now less negative, reaches 0 and became even positive. So we have this reverse. From minus inside, it became positive inside and negative outside, right? So this part over here is called depolarization, right? So we're moving towards plus 30, right? So we're right there. Now what's gonna happen? Now when we reach plus 30, sodium channels now are closed, right? And potassium channels open. But potassium always moving out of the cell. Sodium moves in, potassium out. So now potassium start leaving the cell. So now we're losing positive charges. So we're moving again back to 0, right? So this part over here, and then potassium channels, they even stay open a little bit longer. So we even became even more negative inside. So this called repolarization. So minus 70 resting membrane potential, neuron is polarized. Then we have depolarization. Neuron became positive inside. Then we have repolarization, right, back to negative. And this part over here below minus 70 called hyperpolarization. And then back to the resting state. Right, so here's our big picture. So here's our resting potential, and it kind of give you a key. Now, voltage gated sodium channel, voltage gated potassium channel, and potassium leakage channels over here, right? So now when we are resting, when membrane is in a resting state, all voltage gated channels are closed. What is open? Only leakage channels, right? So that's a resting state. Now, when sodium start moving in, when voltage gated sodium channels open and sodium start moving in, right? Membrane became, instead of being negative, became positive inside. So how we show it on the graph, we go towards positive numbers, right? Now over here, when we go back to negative, now sodium voltage gated channel now closed and potassium voltage gated open. So now potassium start moving out. So we're losing positive charges. How it would look on a graph, we go back to negative, right? And then we need to pretty much get back to resting potential. And it says potassium diffuses back in, but here we also have the sodium potassium pump that's gonna restore our normal balance. So here's the big picture of action potential. We have resting state, we have depolarization, we have repolarization, hyperpolarization, and back to resting state, right? So that's a movement of sodium and potassium across a cell membrane, and that creates this electrical impulse. And when we have complete reverse from minus 70 to plus 30, this is our action potential. And this is how neurons communicate. Right, so over here, again, kind of the same stuff, we're just gonna overview it one more time, and we're looking at a part of axon, right? So an axon, here we try to see what the voltage is. So that's inside, this is outside. So when we have equal concentration of sodium, potassium inside and outside, we don't have any energy, right? We don't have separation of, we don't have chemical gradient over here. So we have sodium, potassium, and the same concentration of sodium, potassium over here. Now, when we separate sodium and potassium, right? Then this is called what? Chemical gradient or ion gradient. So how your cells separate? How they put lots of sodium out and potassium in? This is sodium-potassium pump, right? That's a sodium-potassium pump. So that's a creation of ion gradient. But look, it's still 0 millivolts because sodium is positive, potassium is positive. So we don't have energy over here, right? We only have chemical gradient. But because your cells create this chemical gradient, and because we have leakage channels, right? What leakage channels? Potassium leakage channels. We do have sodium, but again, the membrane is 75 times more permeable to potassium. Now, when those leakage channels are present, potassium start moving down its concentration gradient. And now when potassium moving down, look, membrane became more and more negative, right? Until it reaches -70. And this is our resting membrane potential, right? So here's -70. So chemical gradient, right? That's a chemical gradient. And because of the chemical gradient, we have electrical gradient. So together it's electrical chemical gradient -70 millivolts. Now, when we're moving to plot 30, now we have voltage-gated sodium channels. So we open this voltage-gated sodium channels, sodium start moving in, right? Lots of positive charges moving in, right? So we became inside positive. So this is our depolarization. Now, when those channels are open, potassium voltage-gated channels open, and potassium is leaving, so that back to the resting membrane potential. Right, so that's how crazy stuff is, right? A movement of all those ions create electrical impulses. Okay, so that was kind of like a big picture, right? So I hope it does make sense. If it does not make sense when it's the first time you hear about it, then you just need to, read about it again and then again, and then everything start making sense, right? So now let's look at the steps in the generation of action potential. The first step is initiation. Initiation is arrival of graded potential with sufficient strength to stimulate action potential. Now we're looking at this neuron, particularly this neuron. But what is on top over here? This is another neuron. So that's an axon of another neuron. And this is where we have stimulus, right? So when we have a stimulus, now this neuron releases neurotransmitters over here. Those neurotransmitters binds to their channels and sodium start moving in, right? This is cell body. Now, at the beginning, this neuron is in a resting state, -70 millivolts. Now, when sodium start moving in, what do you think gonna happen? Positive charges moving in, so cell became less and less negative inside, right? So on a graph, we either moving towards 0, right? We're moving up. Cell became less negative inside. Now, this is called graded potential because look, it actually decreases in the amplitude. So when it reaches the beginning of axon, it's called trigger zone. If you measure voltage over here, it's below -55. So it's below threshold. And that's not enough to create action potential. Action potential is generated in axons only. So this stimulus is not strong enough, so it's pretty much dies off. So this stimulus is not strong, so by the time it reaches trigger zone, it's below threshold, so no action potential. Now, what if stimulus was strong? So now here we have a stronger stimulus, right? So again, neurotransmitter is released, sodium start moving in, creating this graded potential, right? So we're moving towards 0. And when this impulse reaches trigger zone, it's above minus 55 millivolts. So it's above threshold. Then that means action potential will be generated. Voltage-gated sodium channels will be open, voltage-gated potassium channels will be open, and we have electrical signal in axon, and this signal will go all the way through the whole axon, and neurotransmitter will be released at the axon terminal, right? So this is initiation. So first we need to apply graded potential that is strong enough to stimulate action potential, right? Next step is depolarization. So let's look at the graph. Okay, where's my graph? No graph, oh, that's too bad. Okay, so maybe, how about, where we gonna get this? I need to add some graphs over here, but we can use maybe this one. Right, so first we had what? We had our initiation, right? So so imagine over here somewhere we have minus 55, right? So if this graded potential enough to reach this minus 55 and above, then action potential gonna be generated. So now if this happen, inside the neurons, we will have voltage-gated sodium channel open and sodium will move in. Maybe we even say over here, right? So if we in axon, and if graded potential that reaches this axon hillock, if it's generating action potential, that sodium voltage-gated channels will be open and sodium will move in. Sodium moves in, and graph will go all the way up to plus 30, right? And this would be depolarization, right? So that's what it says. Depolarization. Local current open voltage-gated sodium channels. Sodium in flux. So sodium moves in. And we open more and more sodium channels, and we have reversal in membrane polarity to plus 30, right? So this is our depolarization, right? So this pretty much is this part, depolarization. Now, what happened over here when we reach plus 30? Well, this sodium voltage-gated channel will shut down, and potassium voltage-gated channels will open. Now, when potassium voltage-gated channels will be open, right? Potassium start leaving our cell. So when potassium leaving the cell, we losing these positive charges, so we go back to minus 70. And this is called repolarization. Here, repolarization. So sodium channels slow in activation gate close. So in activation gate close, membrane permeability to sodium declines to resting level, and voltage sensitive potassium gates open. Potassium exits the cells, and internal negativity is restored, right? So this is repolarization. And then we have hyperpolarization, because those potassium channels remain open longer, allow more potassium to exit, and cell inside became even more negative than minus 70, right? So that's called undershoot. Right, so where's our good picture over here? Right, so this, you know, pretty much also explained everything, right? So you can go back over here. Right, so here's our resting potential, minus inside, plus outside, minus 70, right? Now, this is, membrane is polar, right? So neuron is polarized. This is depolarization, and this is sodium start moving in. So everything that goes up and down, this involved voltage-gated channels. So the sodium moving in, depolarization, right? Sodium channels now closed, potassium channels open, potassium moving out, right? Potassium moving out, so a cell became negative inside again, right? And it's even undershooted, so it's below -70, and then back to resting membrane potential. Right, so that's our, now what is threshold? So if the internal part of the cell membrane reaches -55 millivolts, then it has reached threshold and the action potential will happen. So inside the membrane we have -70. If it ever reaches -55, that's what we call threshold. And that means this electrical impulse will be generated. If depolarization does not reach its threshold, then no action potential will happen. Now, action potential are all or none principle, operate in all or none principle. So if this membrane reaches -55 and action potential happen, it will go along the whole axon. It will not die off, right? So if threshold is reached, we have action potential. If threshold is not reached, no action potential. But it cannot happen that action potential first is generated and then it just decreases in a magnitude and it dies off. No, if we have action potential, it will go through the whole axon to axon terminal, right? So once an action potential has been generated, the amplitude of depolarization wave will be the same for all action potential generated, right? So that means that, look, so this is our resting membrane potential and this is action potential, right? Action potential, action potential. So your neurons generate the same action potential from -70 to +30, right? And so all action potential are alike, and they don't depend on a stimulus intensity. And because all action potential are alike, then how your central nervous system can determine the difference between a weak or a strong stimulus. In other words, let's say if somebody touching you, right, like tapping you. Let's see if I'm still, do I still have you? I lost you guys. Yeah. So, so let's say if somebody touching you, right? So when somebody touching me, what happened? Those neurons, right? I have neurons in my body, in my skin. First, when I touch myself, I open mechanically gated channels. So inside neurons, sodium start moving inside neurons, right? This is called graded potential, right? So graded potential in the soma. So when it reaches axon hillock, right? When it reaches axon, because I touch myself strong enough that I feel it, right? So go ahead and touch yourself and you feel it. So how do you feel this touch? So first you open mechanically gated sodium channel. So sodium start moving inside neurons, right? And so much sodium moves in that your neurons generate action potential. And this electrical impulse moves all the way to my spinal cord and to my brain. So I feel this touch, right? So that's the action potential. So I can feel it because of action potential, because my neurons generate electrical impulse called action potential and it reaches my brain and I feel this touch, right? Now, is it possible that somebody touches me and I don't feel it? That's possible. For example, if you're camping somewhere and mosquito is sitting on your skin and you don't feel it, why? mosquito is sitting, so it's disturbing the membrane of neuron, but its stimulus, mosquito is so, you know, tiny and small, that stimulus is so small that that graded potential that is generated, it's not enough to generate action potential. So you don't feel how mosquito is sitting on you. But when mosquito bites you, that's a strong stimulus, right? And action potential is generated and it reaches your brain, so you feel how mosquito just bit you, right? So that makes sense, right? This is how neurons send signal to your brain. And the same about temperature, same about pressure, same about smell, right? But touch is just easy example. So when somebody touches you, mosquito sitting over here, this is your initial stimulus. If it's not strong enough, then action potential is not generated. So your brain doesn't know that mosquito is sitting on you. Now, when mosquito bites you, right, then it's strong enough stimulus, so it generates action potential and it reaches your brain. You feel it. Now, all the action potentials that reaches your brain are the same, but I can touch myself slightly, or I can really hard touch myself very hard, but the impulse is the same. So how my brain knows what is the intensity of the stimulus? Does it make sense, right? So if somebody nicely touches you, that's a weak stimulus, and I know it. If somebody starts punching me, that's a very strong stimulus, and I know it. But how do I know it if every single action potential is the same, right? That's the question. So how your central nervous system tells the difference between weak stimulus and a strong stimulus if all action potentials are the same? Well, your central nervous system determines stimulus intensity by frequency of action potential, right? So let's say if you have, so over here you have some stimulus, right? So imagine that somebody touches you so lightly that no action potential is generated. So you do not feel this touch at all, right? Right, so this is sub-threshold stimulus. So this is a stimulus, right? You see it goes up. So you do have stimulus, but stimulus is so weak that action potential is not generated, and that means your brain does not feel this touch. Now look, this is a little bit stronger stimulus, right? It's above threshold and action potential is generated. So now if somebody touches you with this strength, your brain feels it. But you see the frequency of action potential? It's not that, you know, the frequency is low. That's how your brain understands this is a weak stimulus. Now here, somebody punching you, they punch you, right? So stimulus is very strong. Action potential is the same, but the frequency of action potential increases, and that your brain knows somebody just punching me, right? So that's a very weak stimulus, or very strong stimulus, right? So action potential is the same, but frequency of action potential increases, and this is what allows your brain to understand if somebody just nicely touching you or punching you, right? So that's the difference. Okay, now refractory period. Your excitable cells, they have absolute refractory period and relative refractory period. So absolute refractory period when sodium voltage-gated channels are inactive. And no matter how big the voltage is, so how strong the stimulus is, you cannot generate new action potential. And relative refractory period, some of sodium voltage-gated channels are closed and some still inactive. So when they close, that means they can be reopened again. And then if the stimulus is very strong, it can generate another action potential. So pretty much you can see over here that absolute refractory period happen, you know, in this period of time when sodium channels are inactive. But over here, you know, the action potential is, well, the cell not back to the resting minus 70 voltage yet, right? So all this area, but some of the sodium voltage-gated channels are already closed, so they can be reopened again if we apply strong stimulus. So we have two refractory periods, absolute refractory period and relative refractory period. During absolute refractory period, new action potential cannot be generated. And during relative refractory period, if stimulus is strong, extra strong, then action potential can be generated again. Okay, so we discussed pretty much. Let's see. So let's just go a very quick review. So we said that cells establish potentials, right? That means they separate charges, and here are some channels that are involved. We also said that cells establish different type of potentials: resting potential, graded potential, and action potential. And action potential, this is how your nervous system communicates. This is how you can feel. It's cold outside. It's hot. Somebody touching my hand. Somebody punching me. This is how you can smell stuff. It's how you can hear me. Your nervous system communicates through this electrical impulse called action potential, right? And generation of action potential involves several steps: initiation, depolarization, repolarization, and hyperpolarization. Now, you also need a stimulus above threshold to generate action potential, right? Now, we also discussed that all action potentials, they are alike, right? So they do not depend on intensity of your stimulus. If stimulus is above threshold, right, that's it, action potential is generated. Now then, how to understand, how your nervous system can understand, is it a weak stimulus or strong stimulus, because of the frequency of action potential that is generated. Now, how fast your neurons can generate action potential? So you can see it's one action potential takes about what, 3 milliseconds, right? 3 milliseconds. Now, can it be faster than that? Well, usually not, because you need these 3 milliseconds for, for your neuron to go plus 30 and minus 70 again, right? So usually that's how, how often your neurons can generate those action potential, because they are in a refractory period. But when they are in an absolute refractory period, that is less than 2 milliseconds, then they cannot generate new action potential. If they in a relative refractory period, if stimulus is really strong, new action potential can be generated, right? Now, next question is, what determines how quickly this electrical impulse moving through your axon? What is conduction velocity, right? Is it moving like fast, slow, and what this velocity depends on? So it depends on myelination. So myelin sheets insulate and prevent leakage of charges. And when axons are myelinated, then conduction is about 30 times faster. So myelinated axons can conduct electrical impulse 30 times faster than unmyelinated axon. And this conduction is called saltatory conduction. When we have saltatory conduction, voltage-gated channels located only at the nodes of Ranvier, and action potential kind of jumps from one node to another. So let now explain, you are looking at this diagram. So if you're looking at dendrites, dendrites are not myelinated. Myelinated means there is some lipid, protein lipid structure covering membrane, right? Cell membrane. So this is cell membrane. And if it's covered by myelin sheaths, then this is myelinated axon. So axon, this axon is myelinated. Dendrites are not myelinated. Some axons are not myelinated. So if we are in dendrite, right? So let's say stimulus is applied, right? But this white part, you see how it's getting smaller and smaller. That means that the voltage decreases, right? So you're losing this charge. You're losing the charges, right? So this is graded potential. Graded potential is generated in dendrites and soma. Now, when you in axon, that will be your action potential. So here again, stimulus is applied. Stimulus it decreases, but then we have new channels open. So we have again sodium in, right? Sodium in, potassium out, right? So we constantly, so voltage does not decay because sodium and potassium constantly moving over here, in and out, right? But the conduction is slow. Now, if we have myelinated axon, right, this myelin keeps this electrical current from leaking out, right? So voltage doesn't decay much. And then the voltage-gated channels are only located between this myelin sheath. So myelin sheath doesn't cover completely axons. It has gaps, and those gaps are called node of Ranvier. And this is what you guys are supposed to learn in anatomy, right? And the movement of sodium and potassium only happen in the node of Ranvier. So this is called saltatory conduction, and it looks like an impulse just jumping from one node to another node. And this is saltatory conduction is 30 times faster. So effect of myelination. So we have myelinated axons that velocity is higher, right? So here we're talking about multiple sclerosis, because what is MS? MS is autoimmune disease that affects young adults. And this is when myelin sheath in the central nervous system became non-functional sclerosis. So sclerosis means became hard, really. Or it can be just, so your immune system attacks your own myelin sheath, right? And it destroys this myelin sheath. And axons that's supposed to be myelinated, now this myelin sheath is damaged, and this reduces the speed of conduction. So your nervous system doesn't work properly, right? So it's a shunting and short circuiting of nerve impulses occurs, and nerve impulse slows and eventually ceases. And because your nervous system regulates every function of your body, then symptoms will be they will vary a lot, right? It will be visual disturbance, weakness, loss of muscle control, speech disturbance, urinary incontinence, right? So that's all the symptoms of multiple sclerosis. And we cannot cure multiple sclerosis. We can just treat the symptoms, and we can hold symptoms at bay, reduce complication, reduce disability, and we can use drugs that pretty much they suppress your immune system. So because that immune system that attacking your myelin sheath, if you suppress your immune system, then less attack and myelin sheath is not damaged that much. Okay, and the last part of this chapter is a synaptic transmission, right? And this happens when action potential reaches axon terminal. So how this electrical impulse goes from one neuron to another neuron, right? So let's look at this diagram. So in this picture, we are at the synapse. Like this is a chemical synapse between two neurons. So here's presynaptic neuron, that's axon and axon terminal, and it synapses with the dendrite of another neuron. So here's our chemical synapse. So action potential reaches this synaptic terminal, right? Now when, so remember, in a normal resting neuron, neuron is negative inside, positive outside. Now action potential is what? Positive charges is coming to the terminal. So it's positive, it's plus 30 now inside. So when this happens, when this electrical impulses reaches axon terminal, it's activate calcium voltage gated channels. Now calcium, so they open, calcium start moving in. When calcium moving in, it causes these vesicles. Vesicles already is there, right? And they already have neurotransmitters inside, but neurotransmitter cannot be released without calcium. Now, when action potential reaches the terminal, calcium in, those vesicles fuse with the membrane and neurotransmitter is released in this synaptic cleft. Now, this neurotransmitter now attaches to its receptor. So here's this receptor, and this receptor is ion channel, ligand-gated ion channel. So neurotransmitter binds, channel open, and what happens? Sodium start moving in. And again, sodium start moving in, right? If you remember the graph from -70, these neurons start, inside became less and less negative. So we're moving towards the zero. And this is our graded potential, right? And this graded potential, because it's in a dendrite, now this graded potential, when it reaches axon hillock over here, if it's strong enough, new action potential will be generated. Now, we do have two types of receptors. So these receptors over here on a postsynaptic neuron, we have two types of receptors, ionotropic receptors and metabotropic receptors. Ionotropic receptor, when neurotransmitter binds to ion channel, and its ion channel allow ions in or out, right? Metabotropic, when neurotransmitter binds to the receptor, but this receptor is not a channel. It will be the chain of reactions that lead another nearby ion channel protein open and ions move in or out, right? So this is metabotropic receptor, a complex that causes metabolic changes in a cell when the neurotransmitter binds to it. So neurotransmitter binds to it, but we still need some metabolic changes before ions can move inside a cell or outside a cell, right? Here, it kind of like direct action, neurotransmitter binds and ions move through the same channel, or neurotransmitter binds to its receptor and then channel next to it will be open because of several intermediate reactions over here. And when this happens, when we have metabotropic receptors, neurotransmitter is called first messenger, and another molecule that will be generated inside a cell and open this ion channel is called second messenger. Right, and here's some neurotransmitters, right? Just for overview, we have excitatory neurotransmitters, inhibitory neurotransmitters. So acetylcholine, for example, is the main neurotransmitter that released between neurons and muscles in neuromuscular junction. Epinephrine, norepinephrine, histamine, glutamic acid. So they mostly excitatory. We do have inhibitory, serotonin, endorphins, dopamine, GABA. Right, so it depends on what kind of neurotransmitter binds to these receptors. We either activate this, target neuron, or we can inhibit this target neurons. That's why neurotransmitters can be excitatory or inhibitory. Okay, so that's it for this chapter. And thank you for watching. And I hope it was helpful.